Hi everyone. So first, thanks to the organizers for the kind invitation and really enjoying the meeting so far. And I know I'm between you and the coffee break, so if you need to wiggle around a little bit just to wake up, that's okay as an intermediate solution. And yeah, today I'm really excited to share with you our progress in this technology section with the dream of really visualizing a drug. In our case, we're focusing on the lipid nanoparticle containing mRNA, especially because I'm hailing from Vancouver, the home of many lipid nanoparticles, as I'm sure you know. And to develop a platform to really follow their dynamics in solution, see their structure, their size, their interactions, and all kinds of cell-like conditions with control, and then mimic the cell-like conditions and follow their journey in cells. So that's really the story that I'm going to bring to you today, that we've been building up over the past several years at the Michael Smith Laboratories in Vancouver. So the motivation of our work is complementary to many of the techniques we heard about today and the ensemble average techniques that are often used in characterization. And perhaps by developing these tether-free, in-solution, yet controlled approaches without pinning the molecules, without tweezing or tethering, but trying to just follow their native motion and interactions, we can bring new eyes into their behavior and then get new insights into their interactions and then optimizing them inside living cells. So that's really what motivates us. And I lead a team of biophysicists. So if you come and visit, which you're really welcome to do, very beautiful rainy weather, just like Cambridge, England. Good time to visit is between, just kind of avoid October to February. It's a pure rain cloud. Other than that, it's pretty good. But we've set up 10 different microscopes now. So our dream is really, could we image that last event of a drug binding to a target? So high-speed molecular imaging of RNA conformation, RNA binding to drug, DNA imaging. But that's not what I'm going to tell you about today. At a more sort of mesoscopic speed, so a few milliseconds a frame, we're imaging in wells, shown here, the lipid particles, which are quite a bit bigger. They tend to be around 100 nanometers, diffusing around, fluctuating, fusing, releasing. That's what we'll talk about today. And then in live cells, where we'll make the connection, we use scanning confocal microscopy. So we heard earlier in this session about really amazing multi-channel imaging. Here we have just a few fluorescence channels and also interferometric iSCAT imaging so that we can look at these cells for a long time. And the work that I'll present today is collaborative with the Peter Cullis group. I'm sure known to many of you, AstraZeneca, and at this meeting as an innovator for over 40 years in the lipid particles that were then used in one of the vaccines, the Acuitas BioNTech Pfizer vaccine that many of us took in the pandemic. So the platform that we've built on is called CLiC for convex lens-induced confinement. Many of us here are physicists, so a lens is any curved surface. And what we like to do is bring together this curved surface with a flat surface so that they're really in the closest possible proximity. So this actually works. We use glass. It's very smooth, and if you squeeze glass together and clean it enough, it will even bond. But what we typically do is mount a little schematic of the instrument, as shown here, just on top of an inverted microscope, looking up. And then there's an artist's rendition of the imaging region that we'll look at throughout this talk, where you're looking up into a thin region that's brought together and trap particles or cells into little wells. So you have many copies of them in solution-like conditions. And they're nicely isolated for quantitative analysis, which is really a big part of what we do. So that's the platform called CLiC. I guess because I'm a physicist, I wasn't quite aware of CLiC chemistry when I named this, so please forgive that. It's named after geometry. But yeah, this is a prototype in the lab, and it's quite easy to use. Before we get into the imaging, you kind of pipette the sample into a little flow cell you can hold with your hand. The sample flows in, very simple. And conceptually, this flow cell is placed in the gadget I just showed. You squeeze the roof onto the floor into which we can pattern any kind of feature. So here are just microwells, a few microns wide, half a micron deep, so the molecules are in focus. And then this is a little video. If you were looking up while you were squeezing the roof down and looking up into some wells. These are just some yo-yo labeled plasmids, around 2.5 kilobase per diffusing. Okay, so the outline of this talk, and it's in the technology session, so I'm going to kind of give you an overview of our vision and a number of papers from 2021 to ones that are just went online, and some in-progress work as well that will go online soon. But at the single molecule level, we'd like to entrap single lipid particles, pull out their size, payload, and even if there's multivalent cargo, be able to really quantify that. So this is, with our collaborators in the nanomedicine community, highly desired, because in their labs they often have kind of a dynamic light spectrum, which is a coarse measure of size from the ensemble, or might have a single particle technique, but with some limitation in dynamic range. So we can go from 1 nanometer little molecule all the way up to hundreds of nanometer using particles and really capture every one. In the second part of my talk, I'll talk about how we're really curious about how particles fuse. So knowing their size, how the RNA is packed, their structure is one thing, but there are a few important reasons why you want to understand their fusion. One is that in the practical sense of manufacturing, you often prepare these in one condition like acidic pH and then transfer to an aqueous pH before you inject. So understanding those dynamics is important, structure formation is important. Also, when we get to following the particles in the cell, the pH drops at the endosome. So could we expect any dynamics? And other groups focus more on this, but the particle fusing to the surface of the cell is another example. And typical methods might use surface plasmon resonance or other interaction cases, but here we'd really love to be able to see and track the individuals as they interact and fuse, and really get at mechanisms of action. For example, what drives fusion and what drives other interactions. And finally, to follow these in cells would be the fourth part. So I hope that if you are curious about this, I just wanted to add some references. There's a few in ACS Nano on the single particle size characterization and our recent integration with iSCAT. If you're curious about the fusion study that I share, it's available on ACS Nano. And I'm going to share some unpublished data that we're just in the process of submitting right now on very in-depth characterization of the mRNA lipid complexes with some neat results. So those aren't published yet, but I hope to spend the time talking you through those in more detail. Okay, so one of our big goals, and this was referred to earlier today, is really not to cook or kill the cells. We saw some nice videos of them dying under illumination. And one of the ways to do that as we build up towards our dream is actually to have label-free imaging. And my background is actually in imaging before moved into biological applications of imaging. And being able to do non-destructive imaging is really at the edge of any imaging field. And recently in the field of biophysics, the technique of interferometric-based scattering imaging, where you shine some light, here we use 4 or 5 nanometers, and look at backscattered light from the sample and interfere it with some of the reference light, allows you to get a signal that is not fluorescent, but yet a signal from every particle and different parts of the cell. So in this story that connects to our early characterization of particles to the more recent work, that's what we're trying to do, get size, payload, mass, and have the ability to characterize with and without labels, and ultimately follow those in cells. So we use the CLiC technique to load the particles in wells, typically at a density of less than 1 per particle. We then track, and we've built very sophisticated simulations that reproduce our imaging data. And from every single particle trajectory in a well, this is a bit of physics that Einstein and Stokes and Boltzmann gave to us. From that long diffusion trajectory in the confined well, you can pull out a very sensitive measurement of diffusion and size. So this is how we can really grab the size of any small molecule or particle here with a label. And we were curious, can we really push this to look at label-free imaging of the particles and also follow them in cells a bit later in the talk. So recently what we've done to enable the step on this journey is we've implemented label-free iSCAT imaging, which involves a reference backscatter beam, as well as multicolor fluorescence imaging, only a few colors, but still very good, on a live cell scanning confocal microscope, which many people have. So part of our quantitation is actually quite accessible to other people now, in the sense that many life cell imaging folks do have these scanning confocal microscopes, far more than the very fancy single molecule scopes we saw earlier in this talk. And then on our microscope, we simply add our prototype of our CLiC instrument, which squeezes the particles into the wells. So the question is, could we characterize size, mass, payload, and do this with and without labels as a first step? So what's shown on the top is, we're curious in general to look at a few colors to characterize the particle, for which we use a DIO, or a dye in that family, which is a very standard dye for lipid particle labeling. We covalently label the mRNA, and we've done excruciating numbers of controls to make sure that these lipid RNA assemblies are not impacted by that label. There are many ways to impact that, and the checks are in cryo-EM as well as in particle-particle interaction. But in the end, even with multivalent cargoes, such as CRISPR, where you might have a guide and an mRNA or even a protein, we can track particles doing iSCAT, multicolor fluorescence, and DIC. So our CLiC wells, this is not the most beautiful set of wells, but anyhow, you can see just by eye here in this picture using iSCAT imaging, very old optical technique actually, just interfering the backscattering with intermediate plane. You can see a little particle diffusing around there, and the wells come out very clearly, and then in fluorescence, a little bit of dye on every particle, you can pick them out. So the signal is really quite exceptional. To calibrate this and this little bit of optics in this method development, we did follow a similar path of the Heath and others who have set up iSCAT on scanning confocal microscopes quite recently as well. And just check that the diffraction pattern agrees with what you would expect. And this is a visualization of the beam focused at the plane. And our very first step was to characterize a few sizes of polystyrene beads from 50 nanometers to 200 so nanometers, 120 nanometers. And this interferometric scattering signal does have some heterogeneity. You're essentially collecting scattering from different light angles. And these beads themselves did have some heterogeneity in size. But this was a very good first control. And if you're curious, this is now very recently published in Nano Letter. Now the first step towards following the journey in cells and a deeper characterization of the lipid RNA complex is this, and we'll return to this a bit later in the talk, is a lipid particle with a little bit of DIO dye, so typically 5 to 10s of those dyes in the particle, not altering their structure, very standard dye, and a labeled mRNA. That took a little bit more work. But you can see that the fluorescence readout from these quite heterogeneous lipid nanoparticles, so these are formulated with the original Onpattro MC3 formulation, the same formulation process that the original therapeutics were formulated with in Vancouver. You can see they're quite spread in size and intensity. This is not a single polystyrene particle, but a biological particle. And you can see the heterogeneity of the light scattering as well as size, but the dependence does agree with what we would expect from theory. So that's the first look. One of the interesting things is that in iSCAT, in addition to the two fluorescence readouts, you can actually quantitate things like mass and water content. So this agrees with what we would expect. And again, if you're more interested in the technicalities of this, we explain how to calculate the water content. But these lipid particles are about 25% water content. And so the next step we did was to add a fluorescence sensor. So we're very interested in the particle's journey in the cell to actually track pH. So as I mentioned, when the particle enters the cell and undergoes eventually endocytosis, it does, you expect the pH to drop. So in another work cited down here, research associate Benju has developed a doubly labeled oligo inside a particle that actually acts as a pH readout. We did a control experiment on our setup and affirmed that as we move from neutral to acidic pH, that pH drop, we see the expected increase in size and swelling. We get the expected range of sizes of these particles and iSCAT signal. So that is one of the baby steps in our vision that we've now established. Another one, in terms of our progress towards comparing these in cells, is to be able, in solution, to actually control the environment. So in the CLiC setup we squeeze the particles in the wells, but there's actually not a requirement to completely seal the wells. I mentioned that we did that before, that's possible, but we can actually leave a little thin gap because the entrapment is entropic. The particles won't actually escape if they're quite a lot bigger than the gap that you leave. It can be, 1 or 2 diameters of the molecule that you're interested in. And think about the depth of the well. It's a few microns wide, half a micron deep to keep things in focus. They're not going to escape. So this allows us to actually trap mixtures of molecules. Here we've stained in red and green. We can follow the tracks of individual particles. And then we can use FRET. So we heard about this earlier, the fluorescence resonance energy transfer between the two dyes when they're co-localized to the particle. So for example, trap a mixture, change the buffer. In this case, we're interested in switching from an acidic to neutral pH and see what happens. What drives fusion? Do they fuse? So this had not been directly shown before. So in solution, when we mix these particles and flow in via the microchannel the solution that changes the pH. Well by well, just a quick visual here, you can see that every little well kind of has a different clock. And in our control experiments, we do measure that wavefront. But in each well, you have a time course of the FRET increasing when this several particles fuse, and the donor decreasing in fluorescence as they fuse. So for every well mixture, as the reaction goes on, you can pull out a rate. So actually in this study, I won't go too long on it, so we have a bit of a survey here. We can look at structure formation during the fusion. And for every reaction, we can pull out a rate. And as a function of trajectory kind of in the phase space diagram, so from initial to final pH, we can vary pH ionic strength separately. We were very curious to get a full picture of what actually drove that structure formation and fusion. So what we did here, which is in more depth in this paper, so we started from the typical condition in which particles are manufactured. We moved by just changing the ionic strength and saw a very, very long time constant, so very, very slow fusion. This is something that our collaborators are interested in because normally just a simple dialysis would be performed. How particles are typically formed is actually along this purple curve with a buffer exchange. And at each endpoint, we looked in cryo-EM at the structures that had formed. This particular graph is with the MC3 ionizable cationic lipid and the Ampatra formulation, so there isn't a lot of structure in these particular particles, but in others there is, and I'm going to show that a little bit later in this talk. But you can see there's a very distinctive on an logarithmic time scale set of rates, and we can carefully measure as a function of pH and ionic strength how the fusion is impacted in this phase space. Okay, so that kind of gives you an idea in this method session of how to look in great detail at these heterogeneous interactions. So the new results that aren't quite published yet that I wanted to share today are actually, I hope, interesting in the context of a lot of recent papers that we're seeing from the Peter Cullis developers group, as well as AstraZeneca and Moderna and many leading academic groups around the world, where folks are discovering that with the mRNA-packed lipid particles, the structure in the particle is not nearly as simple as what I showed cryo-EM. In fact, in many of formulations, the mRNA is kind of poking its head out of the particle like an acorn, or in some papers it's called a bleb. It's developing different names. So we've been curious with our nanomedicine community collaborators for a while, what subpopulations of these particles are actually empty and loaded? What structure do they have? What are the actual copy numbers of mRNA? They're looking quite heterogeneous. And, could we solve the sneaker problem in our optical laboratory with single particle characterization, reaction, cell imaging, and even super resolution imaging? Could we report back size, payload, structure, interaction, and then really try to understand biophysically how that's influencing potency? So in the literature, these blob-associated particles in some cases are being reported to be up to 100 times more potent, possibly because the RNA is in an aqueous environment instead of sort of attached to local lipid environment. So this has been raised as a big question. So what we had to do, again, more technology was required. We've been working on this for a little while. we prepared the mRNA covalently labeled and the stained lipid. And in order to learn about structure in our optical setup, we actually found that we could make some correlative measurements using FRET. So this is a little more complicated optical setup in that we need to rapidly alternate the red illumination, the green and red illumination together, and then the green illumination. So, for example, if your RNA and lipid are pretty close in that distribution of multiple dyes and you get some FRET, you'll see signal for your particle in the green channel and the red channel. And when you illuminate with just the green, you might see some FRET in the red channel. And if you illuminate with just the red, you'll just see the acceptor in the red channel. So we actually found we need to do 3 illuminations. Red and green on, red on for multiple excitations. And then from that signal, we could actually learn quite a bit. So in this study, we're taking quite a bit of data with multiple controls. We were curious to compare this on patch formulation. We say analog, so it's being made over in the Nanomedicine Center in exactly the way it was originally made by our shared grad student, Yao Sheng. And then obviously with ingredients that are purchased commercially, so this is being made at the university with these purchased ingredients, so that's why it's kind of an analog of these named products, which are made by the companies themselves. So this is the Alnylam-like Patro, the Moderna-like, and the Pfizer-like. But anyhow, we use those recipes with academically purchased formulation, and we're just curious, what fraction of the particles are actually loaded? What do their structures look like? What do their sizes look like? And for this, we did quite a lot of experiments, and the data I'm going to show is a little bit complex, so I'm going to walk you through it. So on the left-hand side, every dot is a particle. The blue dots here are deduced to be empty, and the red dots are deduced to be full. What we're plotting on the Y scale is the log of the intensity from the DIO, so the lipid channel. So the first thing you notice is actually the formulations are quite different in fluorescence, and I'm going to show the cryo-EM images in a moment. But you already notice a few things. So on the log scale here, looking at the radius of hydration of the particle, so the size as discerned from diffusion, you see kind of a uniform spread. So with this particular Onpattro formulation, we see very little of those blebs show up. You have kind of a uniform spread in size and quite a large spread in intensity. And somehow, even though there's no correlation here, which is interesting, the particles who are full and have some RNA are brighter. They seem to have more lipid. And since the lipid is just staying proportionally, we think that the lipid packing is tighter in those particles due to the RNA interaction than the empty ones. Whereas if you look over here, and this, when we look in cryo-EM, is more heterogeneous. You see some of this blebbing of RNA coming out of the particles. It's not just smoothly through the particle. The empty particles tend to be a bit smaller. There's a slope on the graph. The full particles are a bit bigger and also brighter in the lipid channel. And then over here, you see similarly sort of 2 clusters with some overlap and a strong correlation between intensity and size. So that's one first thing we can learn. Interestingly, the Onpattro had quite a high fraction of loading. One of our collaborators wasn't super pleased with some other publications in the literature which indicated like 20% loading of their very important product. But our loading fraction is good. It's 83%. That's a good result. And then the other two here, where this formulation is used very, very quickly to address the pandemic with the Moderna-like and Pfizer-like vaccine particle, these loadings are 74, 73%. And noting those weren't optimized. They were used, in the sense that the pandemic was addressed so quickly. But we think those are very good numbers. Now let's get to a little more detail about counting. It's a single particle after all. So we were able to, with our analysis, actually extract quantitatively for the first time the number of RNA from the analysis, and maybe focus on the red histogram. So you can see that with the Onpattro formulation, the one that's not so blubby, going to come to the blebs in a moment and what we can learn about structure from fluorescence data. Most of the particles kind of have around 1 mRNA per particle, whereas over here, most of them have a little more. There is a tail in this histogram, but these two vaccine formulations have a little more, several mRNA polymers per particle. And this is the luciferase gene that we're using here. And so that's quite reasonable. And then we borrowed an expression, and hopefully our paper will be up on the bioarchive soon, but that our collaborators use routinely just when they have a bulk estimate of size, a particular formulation, what do they estimate RNA number? That's what's shown in the black histogram. So that's kind of an empirical formula. But actually, if we look at each of our bars, and since we know size for those particles, and we estimate number that way, our detailed FRET-based quantitation from our image processing actually agrees pretty well with the estimate here and here, but not so much here. So that's actually kind of interesting. For this formulation, you don't quite get the number you would expect. So that's something, if you're in this field, we could always chat about. And then here's the thing we were super curious about. If, as in the cartoon, the dye is spread out around the particle, the RNA might be blebbed or not. And that's this kind of very fascinating thing in the field right now. And if you can see in these cryo pictures, you see a little RNA bleb sticking out of these particles, versus over here in the Onpattro, you don't see that. It looks more like a shell. And over here, you kind of see some nuggets and not others. But we do see one thing we can say. So if you do a little simulation and you imagine that you do have a random arrangement of RNA through the particle, and you do another one where there's mostly cluster, just for a purpose of understanding, you do get a cluster of FRET in these two cases, and the two bleb samples do seem to have two clear clusters. Whereas in the other case, and that agrees with this cryo-EM, you do not really see. You just see kind of the random arrangement predicted FRET and the no FRET, versus in the structured case, you see a cluster where you would expect high FRET. So there's not a perfect one-to-one between these FRET clusters and the blebs, but it is a very strong indicator. And what we're learning is that these formulations are complex. There's not one general rule, but there are some takeaway findings. Oops, and I'm speaking quite a bit. So the last part of our work, this is, so the work I just showed is just what the author's now hoping to submit on the bioarchive soon. And now this last bit is ongoing, and the work of the same individuals I showed before, but the team that's focused more on the live cell imaging. And we've already spoken in this session about these really important questions in the field about tracking the particle endosomal escape. So we've kind of built a toolkit to really study and dissect some of these pieces. But now in live cells, side by side, what we're doing in the lab is imaging model cell lines. These are from cancer cell lines, cultured cells. We can track the RNA lipid, and we can have endosome stage-specific stains. So Yao, a co-supervised student between myself and Peter, has some burning questions about correlating these microscopic findings to what we observed during endosomal escape. And together with a couple, Eric and Eric, who have developed this iSCAT live cell imaging and multicolor on the fluorescence with the real quantitation of these biophysical properties in solution and in cells. I guess we're at the point where we can get individual events, get their trajectories, and start to do this quantitation really in this complex cellular environment of the same properties. And that's very technical, but something we're very, very passionate about reporting back to the field. So that would be the next thing we'd love to report, is to, for those set of formulations we've explored, look at the differential endosomal escape, and especially the differential formulations with these high efficiencies reported with and without blebs in the literature. So just to summarize, we've set up maybe a lab. Someone once told me we maybe solved a sneaker problem where we can characterize in solution size, payload, reactions, journey in cell. And then the big dream, we have set up a robotic arm that we haven't turned on a lot yet, but what we'd really love is to work in kind of that midpoint with our biological collaborators and screen formulations and really help develop that deep biophysical understanding of why some formulations have such higher, 100-fold higher potency than others, if that is something we can contribute. And of course, the upshot is this is all quite heterogeneous. So just to close by thanking many students contributed to the work I just showed, in particular grad student Xiao Zhang, who's joined with Peter Cullis, Jonathan Wang, Martin Jasinski, Eric Boateng, and research associate and postdoc Albert Kamanzi and Eric Olson and others. Yeah, and if you're at all curious to chat, I guess we have a coffee break right now, or feel free to reach out to me. So thanks again.